Hello everyone. Welcome once again to the series CSIR NET Life Science Important Previous Year Question Papers with Solution. Today's topic is Unit 1, Protein Secondary Structure. As we all know, the secondary structures that we see in proteins are majorly alpha helix, beta sheet, loops, coils, beta turn, reverse bends, etc. What we need to know from CSIR point of view are their dimensions, the properties, the different amino acids they prefer, and also the other characteristics. This topic, particulars, protein secondary structure, is also important from other competitive exam point of view as well for life science. So you can have a look at the different type of questions. There might be some slight modification in the question type, but the topic details will also be applicable for you as well. The first question is from CSIR NET December 2023. Which is the latest one. If a length of a single continuous alpha helical polypeptide is 108 Armstrong, which one of the following statement is true? So they have given some amino acids, like how much amino acids this alpha helix will contain, and also the amino acid composition. As I said, the type of amino acids present in secondary structure, which is also called as amino acid propensity for secondary structure, and also the different dimensions, you will have to know about them. Even pay attention to the metric system, like how much is 1 Armstrong, how much is 1 nanometer. So sometimes whenever they are asking some numerical based questions, it will be important to do the unit conversions properly. Now how to solve this question, how to get the solution. First of all, we know that the regular alpha helix dimensions, the one turn of alpha helix, since it's a type of a helix, that means we will have turns in the helix. Now one turn in alpha helix is 360 degree, where it takes a complete turn. The length of one turn is 5.4 Armstrong, and the number of amino acids which is contained in this one turn is 3.6 residues in one turn, right? Hence what you need to do here is, you just need to apply a simple unitary method, like 5.4 Armstrong is the length of 3.6 amino acid residues. Okay. Now the question is asking 108 Armstrong length will have how many amino acid residue? Unitary method means if 5.4 Armstrong is 3.6 amino acid residues, then 1 Armstrong will be 3.6 divided by 5.4 Armstrong, isn't it? And 108 Armstrongs, 5.4 amino acid residues, and 108 Armstrongs will be into 108. So that many amino acid residues will be there, which will come to 72 amino acid residue. Now next is the concept of the type of amino acids present. We all know that in alpha helix, proline and glycine cannot be present, okay, especially because of their structure. There is a constraint that proline residue will introduce a destabilizing kink in the alpha helix because it cannot form the hydrogen bonds. Since alpha helix is stabilized by hydrogen bonds, if proline cannot form a hydrogen bond, it will create a bend in the helix like that, okay, because of the missing hydrogen bond. That will destabilize the alpha helix. So whenever a stretch of polypeptide chain has more of proline residues, then they will not have the tendency to be present in the alpha helix form. Proline is rarely found in the alpha helix, and glycine also occurs infrequently in alpha helices because it is a very flexible amino acid, as the side chain is very small. So because of the conformational flexibility than the other amino acids, glycine, if it is present, it will destabilize the structure. That's why one can be the only answer, because it is saying alpha helix contains 72 amino acids without proline and glycine. Moving on to the next question. This is from February 2022. This is to calculate how many hydrogen bonds will be present within the backbone CO and NH observed in an alpha helix consisting of 15 amino acid residues. Simply put, if you have a 15 amino acid long polypeptide, and if this polypeptide takes an alpha helix conformation, then what will be the hydrogen, how many hydrogen bonds will be required to stabilize the structure? That means you have to know about the hydrogen bonding pattern. These are your options. So let's dig into the solution. First of all, this is the hydrogen bonding pattern in alpha helix. Okay. where you have the CO of I residue. So CO of I residue means the first residue of the helix. See, whenever we have a polypeptide chain, it's not that from the right from the first residue of the polypeptide chain alpha helix starts. No, it's not like that. You may have a certain stretch which is not in alpha helix, then you have the alpha helix, right? So this first residue of alpha helix can't be the first residue of the polypeptide chain. So if we give it a number 1, then it won't be appropriate. That's why instead of calling it as first 1, 2, 3, 4 like that, we will say ith residue. Now the next residue will be I plus 1, next residue will be I plus 2. This is how we will designate the amino acids in secondary structure. If you look at this structure, this is one amino acid as you can see. Okay, let's take this one. So this is the ith amino acid. Okay, this is the first residue. Then this is the second residue which we are calling it I plus 1. Then is I plus 2, I plus 3, I plus 4, like that it is going. If you see, this is the first residue and then this is the second residue which we are referring to as I plus 1. Then the corresponding ones are I plus 3, I plus 4, I plus 5 and likewise. Okay. Now the first bonding is between the CO of ith group. Right. So this is the C double bond O of the ith residue or the first residue. And this is bonded with the NH of the I plus 4th residue. Now we always write, whenever we write a polypeptide chain, we always write it from N to C terminal. Hence, whenever we say in an individual polypeptide chain, that is the C alpha, this side will be NH group and this side will be the CO group, right? So this is the NH of this residue, which is the I plus 4. So from the NH of the I plus 4, we have the connection to CO of I, but we will write I to I plus 4 to maintain the numerical order. Now the next residue will be I plus 1. So you just keep on adding I plus 1 and here it will be I plus 5th, right? So always there is a gap of 4 amino acids in between. Now you can see the second one is the CO of I plus 1. So this is I plus 1, this is the CO, and I plus 5, this is the NH. So NH of I plus 5 is joined with the CO of I plus 1. This is how hydrogen bonding in alpha helix is done. Now if you have a 15 amino acid alpha helix residue, alpha helix structure, let's consider the first residue is ith amino acid and the 15th residue will not be I plus 15, it will be I plus 14. 14 plus 1 because we have considered 1 as the ith residue. So it will be I plus 14th amino acid. Okay. So this is the structure of that 15 amino acid residue starting from ith residue to I plus 14. If you draw all the amino, all the CO and NH groups are there. If you start from here, for example, this residue CO of I and this is I plus 4, this is NH residue. So this will join together. Okay. Then the second one, this one will join with this one. So this is the second one. Like this, you keep on continuing to form the hydrogen bonds. Second bond will be CO of I to NH of I plus 5. Third bond will be plus 1 plus 1. Like that you keep on going. And the 11th hydrogen bond will be joining the CO of I plus 10, which is this group, and the NH of I plus 14. So this is I plus 14 to this group, right? Now, if you have to form a bond, the next bond, CO of I plus 11. So if you take CO of I plus 11, then for this to bond, it would require one more amino acid, right? But the problem is we don't have that amino acid anymore, because this is totally a 15 amino acid residue. That is why CO of I plus 11 doesn't have any amino group to bond with. So, alright, either you calculate this way that you will have a total 11 bonds, or you just remember one logic that the first NH groups, right, first 4 NH groups and the last 4 CO groups, as we saw from the I plus 11, this CO group. So this CO group, this one and this one. These 4 groups, they cannot form hydrogen bond because we don't have any NH group further this side. Similarly, from the starting, this NH group, this NH, then this NH and this NH. These 4 cannot have hydrogen bond because we don't have any CO group before that. That's why these first 4 NH group and last 4 CO groups are not involved in hydrogen bond, as enough residues are not present before and after them to form the bonds. Okay. So when you have a 15 residue, amino acid residue, the first, second, third, fourth, they will have only half hydrogen bonds. Okay, because the first 4 NH groups they can't form. Similarly, when you have 15, 14, 13 and 12, these 4 will have half hydrogen bonds with only their CO groups, with only their NH groups. With CO groups, they cannot form the bonds. And in between 5 to 11, they will have complete hydrogen bonds. So totally you will have 9 between them. And 4 halves means another, 7. And 4 halves means another 2. And here 4 halves means another 2. So total 11 can be there. This is one more way of finding this answer. Our simple thing is that whatever they give the residue, you just subtract -4. That will give you the answer. Because the first 4 is only half, so 2 hydrogen bond is lost there. The last 4 is half, so 2 more is lost there. So you have to subtract -4. Even if they give you a 60 residue alpha helix, so the number of hydrogen bond stabilizing will be 56. So what I have told you is the direct shortcut also, and the reason why it is like that. Okay, so in CSIR point of view, you should know both. You should know shortcuts also to be smart enough to get the answer quickly and accurately, and you should also know the reason behind that. Both are important. Next question is from June 2021. This is a true or false statement. Following statements are made related to protein structure. The first one says hydrogen bonding pattern between the CO and the NH groups are N to N plus 3 in alpha helix, N to N plus 4 in 310 helix, and N to N plus 5 in pi helix. Now this is right away wrong, because just now we discussed that in alpha helix we have N to N plus 4. N, if you are getting confused, we can write instead of I, we can write N also. Okay, so this is another notation for writing the number of residue. Whatever it is I to I plus 4. So this is right away wrong. A is wrong, right? Here you have to get a combination of statements. Now in a beta turn, there are 10 atoms between hydrogen bond and acceptor. In a gamma turn, there are 6 atoms between hydrogen bond, acceptor, donor and acceptor. So let me tell you this concept over here about the turns. See, this is a stretch of amino acids. Okay, so this is the ith residue, this is I plus 1, this is I plus 2, because these are the peptide planes, planar peptide planes, in between which the C alpha atoms are present. Okay, so likewise this is I plus 3 and this is I plus 4 and this is I plus 5. So when we have an alpha helix, I have told you the I plus 4 NH group. So this is the I plus 4, so NH group will be this one, right? And this is the ith group, so CO group will be this one. As you can see, there is a hydrogen bond connecting CO of ith residue to NH of I plus 4 residue, right? Now just look in the backbone of this hydrogen bond. That means don't take these atoms, okay? Take the atoms which are in the backbone connecting from the CO to NH. How many atoms are there? You will find a total of 13. This is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 and 13. Starting from the O till the H of the NH. Okay, you didn't take the other non-bonding atoms away from the backbone. So total 13 atoms are covered in the backbone, and as we know in alpha helix, in one turn there are 4 amino acid residues roughly, and the hydrogen bonding pattern is also 4 amino acids away. That is why sometimes alpha helix is referred to as 4-13 helix. Now by the same convention, if you have written 3-10 helix, so you can well guess how many amino acids will be there here, what will be the hydrogen bonding pattern and all, right? So 310 helix, you have the hydrogen bonding from I to I plus 3, not I plus 4 as in alpha helix. Okay, so here the hydrogen bond is 3 residues away. In one turn there are roughly 3 amino acids, and most importantly there are 10 atoms in the hydrogen bond backbone. So for 310 helix, the bonding is still here. Okay, so CO of I to NH of I plus 3. Here if you see 10 atoms are covered. Now another important thing is, whatever hydrogen bond we find in 310 helix, it is same hydrogen bonding pattern we find in beta turn also. Both are different types of secondary structure. One is a helix like this, another one is a turn which is stabilized by 4 amino acids. Okay, so the first and the fourth, that is I to I plus 3, they will hydrogen bond. Even in 310 helix also hydrogen bonding is same. That is why same number of atoms will be present in the backbone, which is 10. So the statement which was saying beta turn has 10 atoms in the backbone was in fact a correct statement. Okay. Whereas what we have 2.27 ribbon. So 2.27 ribbon is also a type of helix in which the connection is I to I plus 2, CO of I to I plus 2. It has 2.2 residues in one turn, hydrogen bonding pattern. 7 atoms are there in the backbone. So this is similar to a gamma turn. Now a gamma turn has 3 amino acids where the first, second and third is present, and the first and the third are in hydrogen bonding, that is I to I plus 2, right? So hydrogen bonding gamma turn will also have 7 atoms in the backbone, but it was written that gamma turn has 6 atoms in the backbone. That is why it is a wrong statement. Got it? Now regarding the parallel and anti-parallel beta sheet, so they have said that parallel beta sheets have evenly spaced hydrogen bonds, but they are present at an angle. So as you can see in parallel beta sheet, you have evenly spaced, but they are non-linear hydrogen bonds. That's the reason why the parallel beta sheet is an unstable structure, as the hydrogen bonds are non-linear. Okay. Whenever the hydrogen bond, that is the DH and the acceptor, donor and acceptor of a hydrogen bond, they are in linear attachment with each other, linear orientation where the angle is 180 degree, then they are very stable, as we see in anti-parallel beta sheet where you can see the hydrogen bonds are linear. That is why here the first and the So the first statement is wrong and the C statement is wrong. They have asked correct statements, hence the correct answer over here will be B and D. Okay. I hope you are getting how important different details are. One where dimensions are important, another question where hydrogen bonding is important, another question where turn orientations are important, right? And there is another question which is regarding some other secondary structures. So for example, collagen. We all know this, so this question is from September 2022. We know that collagen is a type of protein whose structure is mainly secondary only. Okay. So they have a different type of structure which is a triple helix. 3 polypeptide chains are connected with each other in a helix, and that forms a tropocollagen, which is the unit of collagen. It has a triple helical domain structure which consists of 3 distinct alpha chains. Now collagen triple helix is stabilized by isoprenyl bonds. That is not correct, because first of all, collagen, they have this alpha chains. These alpha chains are not alpha helix, okay, because alpha helix is right handed, but these alpha chains are left handed. Again, a detail concept. Okay. So because this question keeps on coming regarding collagen, in some other question they have said that right handed and left handed. So 3 alpha chains which are left handed orientation, they will join together to form a right handed orientation collagen. So the super helix that is created is right handed, but the individual alpha chains, they are left handed. Remember that. And between the alpha chains, yes, you have these hydrogen bonds which are formed between the alpha chains. So collagen triple helix is stabilized by hydrogen bonds. Okay. So B is wrong. Yes, so as I told you, each alpha chain has a left-handed polyproline type 2 helix. That's correct. So just now I said that proline cannot form hydrogen bonds. That is why if you have a helix consisting of many prolines, that is a polyproline type, then they can't form hydrogen bonds. And even alpha chain doesn't have hydrogen bond in it. It is just alpha helix. And that is the reason why this statement is correct, that each alpha chain has a left-handed polyproline type 2 helix. Each alpha chain is composed of multiple triplet sequences of glycine, Y and Z. So actually the glycine content is very high in collagen residues, in collagen polypeptide chains. Okay. And then the next residues which are also found is proline and lysine, and specifically the proline is having a modification where it is hydroxylated at some carbons, and that's the reason why the triple helix structure is even more stabilized. So this is a modification done to get structural stability, right? So even D is correct statement. So you have to mark all the options. Obviously here A is correct, D is correct and C is correct. So first option is the right answer, right? So this is the structure of collagen. As you can see that the content of glycine is a lot more. In fact, every third residue of collagen structure is glycine. And how are these glycine residues, how are these polypeptide chains synthesized? So as you can see, they are synthesized as this alpha chains. Another detail that I will give over here is generally collagen structure will be composed of 2 alpha 1 chains and 1 alpha 2 chains. Okay, this might be useful for some other question. So this is again a correct statement. Generally they will have a N terminal and a C terminal propeptides which will get cut off when they enter inside the lumen of the endoplasmic reticulum. So C terminal propeptide initiates triple helix formation, and procollagen is secreted into the extracellular space, where you can see that the propeptides are cut off, and once they are arranged into triple helix, so they will be released as tropocollagen. Later the tropocollagens, they will again cross-link to form the proper collagen. So you can study about the collagen biosynthesis also, which is also an important part of the CSIR exam. Okay. That's all from now. Let me know in the comment section if you found this video helpful, and also let me know which topic you want me to discuss next for this particular series. Thank you everyone.